Welcome everybody to our today's webinar on Bio-Geo-Chemical-Interfaces investigated with microsensors and 2D imaging. I'm Robert Meyer and I'm a chemist by training. I'm responsible for imaging techniques at our company VisiSens, and with me is Dr. Martin Gutbrot, who is responsible for microsensor measurements and microprofiling techniques. Today we're going to talk on, first of all, the importance of bio-geochemical interfaces and what kind of processes take place in there, but also two measurement techniques from our company, microsensors and 2D imaging, how they can be used for investigating those interfaces, and then we will hand over to some practical examples rounded up by a question round. Please let me first start with some words about our company VisiSens. We are based in the south of Germany in Regensburg. We have been established around 20 years ago as a spin-off from the local university, the University of Regensburg. We are a privately owned company and we are specialized on monitoring the essential parameters oxygen, pH and carbon dioxide. Currently we are more than 90 employees and we offer certified quality due to ISO 9001 as well as ISO 13485. Our product range covers the full range of optical chemical sensors for oxygen, pH and carbon dioxide. And lately we also added the parameter biomass to our product range. We have a pretty diverse sensor applications or sensor formats that we can deliver. For example, non-invasive sensors that allow you for measuring through the transparent wall of, for example, vessels or glass vessel or plastic materials. And we also offer micro-invasive sensors such as our microsensors, but also sensor probes.
Hello everyone. I'll take over and quickly give you a short introduction of why we actually think it's important to study biogeochemical gradients, or in general microgradients. Coming to application fields and samples where those microgradients are actually used. Typical examples include samples like microbial mats, biofilms, microbial cultures, soil and sediment, as well as plant and animal tissue. As you can see, actually the variety of those samples is quite diverse and quite heterogeneous. But what they all have in common is that usually microgradients establish within their boundary layers on a very, very small range. So we are always talking about the micrometer to centimeter range where actually stark gradients can be observed. Why is that the case? Most of these samples actually constitute semi-solid substances. And some of those, like for example here a sediment example, they are also diffusion limited. So the simple fact that we usually observe physical parameters like light or temperature also leads to establishment of biochemical parameters like CO2, oxygen, mineral or nutrient gradients. And as we're talking about complex heterogeneous systems, these gradients usually don't stay constant. They vary spatially, for example, in the lower zones of the sediment to the upper zones or from left to right, but they also vary temporally quite strongly, for example in day and night cycles or over the year or due to changes of any kind of fluid mechanics, which we can see here. So this strong change actually it also requires techniques that are able to capture those states and also those changes, because they depict or they're actually a representation of the biological system, and those areas actually like the surface sediment or tissues are among the most biological active systems that we know of. So there's a strong need for high-resolution measurement techniques. We will introduce two of those techniques today. On the one hand, we would like to talk about microsensors, When we talk about microsensors, so our understanding of microsensors, we actually mean very, very fine sensors. In this case, they are made out of a glass fiber with a sensor coating on the very, very tip. And this sensor coating consists of a fluorescent dye that is sensitive to the oxygen concentration. So by the excitation of this dye, we get an emission spectrum back, which correlates to the actual oxygen concentration. Similar technique, but in a different format or shape, is the 2D imaging. In the 2D imaging, actually the same fluorescent principle is used, but in this case the fluorescent dyes are actually embedded in a two-dimensional foil. So the foil, for example, would be here along a transparent wall, for example inside an aquarium or a sediment core, or it can be any kind of transparent vessel. And then this foil actually detects the oxygen concentration from the sample in its vicinity, in its diffusion vicinity in 2D. And at the very back we've got a detector unit that can do a two-dimensional readout of this foil here by the same principle, by the excitation and then by the emission of light back from the foil. What those two techniques have in common is that they can all be applied on live samples. They are only, they are even non-invasive or only micro-invasive. There is no need for fixation of the samples, so meaning we can really study active biological systems without need of killing or fixation of the sample. Okay. Explaining a few more backgrounds about the actual measurement principle. As I just said, all our technology, so everything Presense offers, is solely based on light measurements. Everything is optical, so there's no current, electricity, voltage measurement involved at any kind. Here's an example of our sensor spot technology. In the sensor spot technology, the sensor is in the form of a small dot or spot that is glued to the inside of any transparent wall. From a beaker to an aquarium, it can be anything. Then we've got a light guide coming from the outside who does the same. It sends some light onto the spot. We get an excitation back, and this light that actually is being reflected back, we then can detect and correlate to the actual oxygen concentration. The measurement technique itself in case of oxygen is based on fluorescent quenching, which also means that there's actually no consumption of the analyte at all in the measurement involved. Which brings us now to the benefits of chemical optical sensors. As I just explained, it can be non-invasive, which is very nice, for example, for respiration measurements. In respiration measurements, you usually have enclosed system that you don't want to disturb over longer periods of time. With this technique, the imaging or the spot technique, you can just leave it closed and measure from the outside through the wall whenever you like. Another application where it's really important that it's non-invasive is in sterile applications, for example inside a bioreactor here, inside culture media, cell cultures, whatever. It also allows you to integrate our spot first, then disinfect your sample and then leave it running for extended periods of time without need for opening or without a need for putting in a larger macro probe. As I just explained, we are based here on fluorescent quenching, so we have no analyte consumption, which also leads to the fact that actually the measurement of the oxygen concentration is independent of the flow velocity itself. This is also a very nice thing. And last but not least, we have got no effect from electrical interference. For example, you can see over here that the work is actually performed inside an incubator, but actually any kind of electrical measurements you have any kind of electrical device you have around, like a cooler or a shaker or water pumps or lights or whatever, they don't affect the measurements at all, as we solely measure light. So coming back to the first measurement principle, a little bit about the background itself for the microsensor. The microsensor you can see here in our automated micro-profiling setup is actually only this very, very small blue needle here. It has got a really high spatial resolution and can have a tip size down to 30 or 20 micrometers. But in order to actually to also use and to apply this very high spatial resolution, you need very precise mechanics to move the microsensor in such a small spatial scale. For this we have the so-called automated micromanipulator, which is a linear stage here that can move the microsensor in one dimension in a resolution of down to 2 micrometers. So it's really very, very fine scale measurements with very high resolution. To be able to do this, you will need, of course, a very stable setup. You will need something that doesn't move and that's vibration free. And this we achieve by the so-called heavy stand. It's simply a very, very solid base plate that allows you to mount the microsensor and the automated micromanipulator in any dimension, so on the side, on the back, wherever you like. It weighs around 15 kilos, so about 30 pounds. It has really high inertia, high mass, and thereby prevents actually most vibrations and disturbances from the outside. Then you will need the so-called transmitter. It can be one of these handheld ones like here, or one of our smaller versions that we now offer, which uses or reads out the microsensor's signal and translates it into a digital signal for the computer. And then you need a computer with a software who integrates, on the one hand, your depth measurements from your motor and your concentration measurements from your microsensor. Talking or speaking quickly about the different microsensors that we actually offer, and there are pictures up here. So we got 4 different builds of microsensors in general. The first build would be the so-called profiling microsensor. As the name indicates, it's used for most profiling application. Why is that? Because it's the most robust sensor that we actually offer in terms of the actual fiber tip itself. It's housed inside a stainless steel needle. It can be extended, and it's very rigid and solidly placed inside the steel needle. So, which leads to the fact that it's actually very robust in a vertical axis, and yeah, it does withstand profiling into sediment and soils, of course, to a certain extent, but yeah, it's quite robust inside the steel needle. The next sensor that we have is the so-called needle type microsensor, so needle type housed NTH sensor. This sensor is placed inside a syringe with also a stainless steel needle, and a sharp one in this case, which is usually used for penetration studies, for example, inside of packaging or inside of vials or septums. You can use it to pierce through, and then you can extend the fiber tip itself by the plunger mechanism of the syringe. The third type of sensor that we have is the so-called NFSG, which stands for fixed needle type sensor. Why fixed needle type? Simply because the fiber is actually not, it can't anymore be moved inside the syringe, but it's actually fixed. So it's glued inside here to a fixed position, only extending a little tiny bit, a few microns inside the cannula. Why would you need that or want that? In some application in penetration studies, so for example a septum or a vial, you also need the sensor to be gas tight, and this we achieve by gluing the fiber into this needle, so actually no gas exchange can happen between the outside and the inside. This is especially important for anoxic studies. For example, if you study processes like denitrification or anamox, you really want to be sure that no oxygen can penetrate inside your sample at all. And last but not least, we got the so-called implantable microsensor, the IMP microsensor. This is simply the bare fiber itself, the bare glass fiber coated with our sensor technology at the very tip. And it's usually used for custom-made integrations. For example, if you've got an own flow cell or microfluidics or whatever, it's simply the bare fiber that can be integrated by yourself into your system. Talking a little bit about the sensor specifications, first of all, I would like to talk about the measurement range. So for oxygen, we are on the level from 0 to 100%. But as said before, for hypoxic or even anoxic studies, we actually also have a trace oxygen sensor. This is important, as for these anoxic processes like denitrification, you really want to know if you got 0.1% of oxygen or 0.0001% present. And therefore we have an own special sensor for this very, very low range to get enough resolution that you can see anoxic and hypoxic processes and changes. For the pH sensor, we are talking about a pH range of about 5.5 to 8.5. The pH range that you can use it is actually larger, so you can measure down to 4.5 and up to 9.5. But as our sensor response is not linear, the lower ranges and the higher ranges actually have a decreased resolution. But the sensors in general work along this range here. For CO2 sensor, it's still in the prototype stage, but we also got a CO2 microsensor that measures on the 0 to 5% to about 5,000 parts per million. And the resolution of that sensor is around 50 parts per million down to 30 parts per million, which is usually sufficient for atmospheric CO2 concentration and for ocean acidification studies. Coming to the response time, we have two different kinds of sensors, the TS sensor and the so-called TF sensors. I'll explain that in a second, what's the difference. The TS sensor for oxygen can be really fast, below 3 seconds, usually even faster than a second on the milliseconds level. And the TF sensor is actually also very fast and below 10 seconds. For pH, we are talking about a response time below 30 seconds, and for CO2, it's below 10 minutes. As I just mentioned, there's two different builds of the microsensors. So on the one hand, we got the so-called TS sensor, which stands for tapered sensor tip, meaning the tip of the sensor is actually thinned out at the very end, and it can have Okay, as I just explained, we have got two different tip sizes. So the so-called TS sensor, which stands for tapered sensor tip, meaning the sensor itself has a very thin tip that is thinned out at the very end. It goes down to 20 or 30 micrometer, and the TF sensor, which stands for flat broken sensor tip, has a diameter of about 140 to 250 micrometers. And the TS sensor is the faster one, as explained before. So coming to the minimal step size, the minimal step size actually refers to the motor control or to the manual control, and this is as small as 10 micrometers. So 10 micrometer is the smallest interval or step size that you can move the motor inside a micro profile. Regarding the temperature range, it's pretty homogeneous from about 0 to 50 degrees. Only the pH and the CO2 sensors, as you can see here, they have a slightly higher range. This is simply due to the fact they still work in the very, very low temperature ranges, close to freezing, but they get very slow. So from 0 to about 5 degrees, actually the sensors become much slower than that. So you will need to think about if you can still apply it for your application or what the dynamics of your system are. And last but not least, where can the sensor be applied? So our oxygen sensors can be applied both in air as well as in aqueous solutions. It's also possible to use them in ethanol or methanol, or if you need to clean them. It's also possible in 100% ethanol or methanol. For pH and CO2, they both need to be applied in aqueous solutions so they don't work in a dry environment. And for cleaning procedures, you could also use ethanol and methanol for pH. And the pH range where you can use the pH and CO2 sensors is restricted to about 2 or 4 to pH 9 and 10 respectively. Okay, a few words about the software itself for the automated profiling system. As I mentioned before, it gives you on the one hand the concentration measurement here, and then it integrates it over the depth measurement that you have performed. It will give you a profile graph with the different areas and replication and also annotations and everything you can set during a profile run. But it will also allow you actually for two types of view here. So on the one hand, the profile view, but you can also pause or stop your profile in the middle here or at any kind of interesting region and actually start a time series recording down here. So there's a second window you can always use to just monitor dynamics at a certain place or depth in your profile if you're interested in that. The software is now also compatible with all new generations of presence transmitters, meaning that you can use them as well with the microsensors I just explained, but also with the dipping probes. For those of you that don't know our dipping probes, the dipping probes are essentially larger macro probes, steel probes that are much more robust with a diameter of about 3 millimeters to 6 millimeters, and they can be applied in very coarse and rough substrate, like for example carbonate rubble or some sediments with shell and other larger rocks inside where fear of breaking a microsensor, you can easily use our dipping probes. Also what you can do with the software is you can set individual profile and step zones. This is actually shown here. So a profile is performed in 3 different zones. And all of these zones actually have a different profiling step zone. So for example, in the zone where not much happens, zone 1, we choose the rather large step zone. In the zone 2, in the area of high change, we used very fine scale profiling, which was lower than 100 micrometer steps. And in the last zone, zone 3, we used intermediate step zones. This you can define as you like and as applicable to your sample. And last, I would like to show the live video monitoring function of these microsensor measurements. And those of you that have worked with microsensors will know that it's sometimes really hard to know where exactly in your sample you are measuring. This is why we included this live measurement, this live video function. You can use either an endoscope or a small USB microscope. An endoscope, you can even go inside the water and then monitor your sensor tip in very high resolution. It saves you hopefully the need to work with a bulky stereoscope or also to have to work under a microscope. And next we come to a short introduction about the basics of the 2D imaging principles, and for that I would like to hand over to Robert.
Thank you, Martin, for introducing us into the microsensors and microprofile measurements with the micromanipulator systems. I now want to show you or give you some words and hints about our 2D imaging technique. 2D imaging means that we are recording images, so we get two-dimensional information of analyte gradients or analyte distributions. Martin already explained that the sensor foil is usually in contact with the sample, and then can be read out with a detector unit through a transparent glass wall or also directly. So what you do is you take a picture of the sensor foil with the VisiSens detector, and thereby you get an Analyte map across your sample. And you can either way measure through a transparent glass wall, or you can directly place the sensor foil onto your sample and record an image with the detector unit. For the VisiSens systems, also three types of sensors are available: oxygen sensors, pH sensors, and carbon dioxide sensors. The measuring range for the oxygen sensors is 0 to 100% air saturation. For pH, we have two different ranges. One for the more neutral or physiological range with a maximum sensitivity between 5.5 and 7.5, as well as one pH sensor for the lower region between 2.5 and 4.5. For CO2, we also offer two different kinds of sensor coatings, one from 0 to 1% CO2 partial pressure and one from 1 to 25%. The response time of the sensors is approximately 30 seconds for oxygen and pH and lower than 3 minutes for CO2. If you work in the gas phase with the oxygen sensor, the response time gets less than 10 seconds. The sensor foils usually have a size of 4 × 4 square millimeters, as you can see above here. These are the sensor foils which are usually sold with the VisiSens AX devices for medium and micro scale measurements. And they can be cut, for example, into any kind of or any desired shape or size that you need, just by using scissors or scalpel. For our new VisiSens 2D imaging system, which I will refer on later on, we also have bigger sensor foils up to 10 × 15 square centimeters. The number of sensor points or sensing points that you record within one data set, that means one recorded image, is more than 300,000 at a time. And the sensors work in a temperature range from 5 degrees Celsius to 45 degrees. The VisiSens detector comes with a software that allows you for recording single images, but also allows you to do automated time series measurements where you can choose intervals from seconds up to months. What you get is a pseudo-color representation of the two-dimensional Analyte information where each color corresponds to a certain Analyte value, and then these images can be used for analyzing and calculating the values. What you can also do is you can do some multiple views where you can see evolving things during a time series, or you can do one-on-one image comparison where you, for example, can compare the first image with the last image of your time series to see what changed or what happens in between. You can do alpha blending to see underlying structures, as well as freely choose your regions of interest. So usually you have your image recorded, and then you can choose a region of interest that you're interested in, for example some metabolic hotspots, etc., and then you can do statistics with these. So you can do, for example, histograms, but also get average values and standard deviations, etc. And you can also do time series profile plots of various regions in your data sets. So now we will hand over to the practical examples, which is, as I guess, the most important part of the webinar. I can here point out that there are some lots of application notes on our website, www.presense.de, that you can find there in our knowledge database. And today we will focus on some microsensor measurements and 2D measurements at Bio-Geo-Chemical-Interfaces. And I also want to tell you that you can ask questions during the webinar, and we will answer them after the presentation, so please feel free to ask if you have any questions or if anything comes up to your mind. Now I will hand over to Martin again for some nice applications with microsensors.
Thank you, Robert. Okay, enough of the theory. Coming to some real practical examples where microsensors are applied, how would it look like? We see here in a case where the microsensor actually penetrates inside the soil, principle, again, it measures here at the very tip only. It detects the oxygen concentration, and by the emission spectrum back, we can directly correlate. Exactly. Coming to our first example, it's an example from tissue engineering. In this case, oxygen gradients were measured in collagen 1 gels. These collagen gels were in 96 or 24 well plates, respectively. And then the automated micromanipulator system was used to actually profile here, first in the air, then inside the growth medium. In this case, it was stem cells, adipose derived mesenchymal stem cells, through the growth media, and then inside the gel or scaffold where the cells were placed or loaded. Here's a short image on how it happened. Of course, it's an exaggeration. In general, the microsensor tip size is much smaller than you see it here. But in this case, the microsensor was moved first through the culture medium and the air, and then into the scaffold where actually cells, stem cells were present. And these, of course, they respire, so they consume oxygen inside the scaffold or gel. And as we are also talking about a diffusion limited area here, it's quite likely that actually oxygen gradient develops inside the scaffold. Coming to the results we see here, so it was after 5 days of incubation in vitro. To show you in the graph, here we're talking about 1, 2, 3, 4, 5 millimeters. And actually the surface of the cells, so here we are talking about the phase in the medium itself, and here we are talking about the surface of the scaffold or gel, which was loaded with cells. 3 scenarios. So in the first scenario, the control, there were actually no cells loaded inside the scaffold, so the scaffold or gel was empty but present. In the second scenario, it was loaded with an amount of stem cells, and in the third scenario, the amount of stem cells was actually tripled compared to the first one. And we can nicely see here that actually within the medium, the oxygen concentrations are quite constant. Also for the control, we see that actually not much is happening inside the scaffold. So the scaffold is almost on the same level as the oxygen concentrations from the medium, seeing that the scaffold is mostly inert in terms of oxygen, not contributing to any kind of the respiration effect we are seeing down here. And the 2 graphs here clearly show that already within few tenths of a millimeter, so already within 1/200 micrometer, we can see a strong decrease in the oxygen concentration here, which is directly proportional to the amount of stem cells present. Why is that important? Why would anyone like to know or to monitor that? Very simple. The main aim in these in tissue engineering is to actually to grow those stem cells, and the growth of the stem cells depends a lot on the oxygen concentration present. But it's not only the growth; it's actually also the differentiation capabilities of those stem cells. So, meaning stem cells need a certain oxygen concentration to differentiate into certain types of tissue. And thereby in culturing and growing them, it's very important to be able to keep them at a certain oxygen level. And for that, of course, you will need those micro techniques we are presenting here today to actually monitor and track those conditions, to be able to create a system that can keep it stable and constant for extended periods of time. Coming to our next example, which is very different, but also a really nice example. At this point, I would like to say thank you first to the UFZ in Magdeburg. It's a cooperation between Presens and the UFZ that is currently taking place, where we look at river ecology and at biofilm formation inside a river and inside artificial flumes. What was done here is actually a river or stream was redirected through a specially built container and through flume chambers. As you can see here, why was this done? Simply because inside the container the light as well as the flow rate and other parameters can be kept constant. So it can be kept constant, and this is why we can study the biofilm formation with actually most of the parameters well known and well defined during an experiment. The biofilm formation was monitored under different flow heterogeneities or topographies. To do that, actually the bed of this flume was embedded with glass beads. In this case, we've got very, very fine glass beads. You can't see them anymore because they're actually already completely overgrown by biofilm, by sediments, by detritus, by algae. So it's a whole microbial and probably also eukaryotic winter wonderland in there. Everything is present, which already covered those glass beads, those very, very small glass beads. Then we had different scenarios of glass beads to create certain surface roughness. You can see larger ones, we had even larger ones, and then the last scenario was actually with normal rocks taken out of the creek or river to see how biofilm formation was happening here under regulated flow conditions. Coming to the results, how was it done? So as you can see here, we used the microsensor, and the flow direction was in this case from the left, to actually map or monitor across the topography of the stone here. What you can already see very, very nicely is that first of all, the microsensor is really micro and minimal invasive in this system. So it does not, it's so fine, it's got such a small tip, it does not disturb your biofilm at all, and it also does not disturb the flow regime. The other thing that is really nice is that you can use it as well to map across heterogeneous topographies, meaning you don't need a flat surface, you don't need a transparent wall or glass to measure through. You can just measure randomly actually everywhere you would like along those stones, and therefore set your sampling design randomized or as you would like. Showing the results, we can see here mapping from the direction here towards the flow direction of the stone. We can see that at the very, very back, gradients develop much, much stronger from almost saturation in the overlaying water down to almost anoxic or below 10% of oxygen concentration. At the very, very back here, this was due to detritus forming and also to thicker biofilm and sedimentation at the back compared to the very front. The more we go to the front, the more oxygen ingress we can observe, which was due to the thinner biofilm as well as to more algae being present here, and thereby also photosynthesis taking place. If you have done such a profile series, what you can do next is actually use the profiles themselves to perform so-called flux measurements by actually using linear gradients, and by applying Fick's first law of diffusion, you can then use to actually calculate fluxes in and out of your two-dimensional system. Or if you do volumetric integrations, you can use it to calculate activity coefficients. And with this, I would like to come to the last example, which is, I believe, a very typical or often applied case, is to use microsensor inside of sediment cores. Here I want to point out mainly two different advantages. So first, you can use a microsensor actually to profile continuously on the same spot. If you do such an experiment or such a sampling in a perturbation experiment, you can then monitor your changes over time. For example, if you do an addition of nutrients or minerals, or if you do an ocean acidification study, you can see how oxygen, pH, or CO2 would change over time at a certain spot. But as said before, the microsensors furthermore also allow you to actually select different regions, which we see here. So we got this profile region 1 here, where it was continuously profiled over about an hour, and the profile set here is also taken over about an hour course while we were doing the perturbation experiment. And what can be seen nicely here is that you actually have a really high spot-to-spot variation. So you can use it nicely to actually profile around your sediment core, and thereby, for example, you can clearly say that actually the differences or changes observed due to your perturbations are much less than actually the spatial variations. So you can observe within the core itself. And by doing a randomized design, nice statistics can be applied. With this, I'll hand over to Robert again, who will give us some really nice examples on the imaging.
Thank you, Martin. Now we'll head on with examples, exemplary results, application examples for the 2D imaging systems. So first of all, I want to start with some exemplary results that you get by our imaging systems. And here you can see again one of the imaging systems imaging a sensor foil in here in a small Cora. These guys in here are the VisiSens AX detectors. They are available for oxygen, pH and carbon dioxide, and they are USB plugged cameras, and they are used to read out the sensor foils. What you first get is like a two-dimensional readout with the colors in each pixel represent a certain Analyte value, and you can do either way the one-shot measurement to see on what's going on in your sample, or you can also investigate it in a time series and, for example, play like a slideshow and see what changes and which is the most important region that you want to investigate in. Furthermore, then you can select a region of interest where, for example, you have some hotspots detected or you have some action going on in your system, and then you can freely select the region of interest and plot the Analyte values over time. And you can do that several times within one data set to compare, for example, different regions. And further on, you can also measure gradients. This is also one of my first examples, a gradient measurement at the sediment water interface with a Cora sample. In here, in this image, you can see the sediment Cora with the sediment sample and some seawater right above it. This was equipped with an oxygen sensor foil in the inner wall or on the inner wall side of the sediment Cora at the sediment water interface line and imaged with the VisiSens A1 detector. So you typically get your two-dimensional pseudo-color image where you can already see your sediment water interface line and some steep gradients from the highly oxygenated water phase and the poorly oxygenated sediment phase. What you can also see in here in this image is that you do not have an even gradient over the side wall because you always have some spatial variations depending on where you measure your profile. So you can choose, for example, several profiles, get several plots out of it, and you can do then statistics or over time investigations. My next example is an example on, again, in the sediment Cora on benthic disturbance recovery dynamics. In this case, an intertidal sediment sample was taken with the Cora, put into the lab, equipped with a pH sensor foil, and then recorded with the VisiSens A2 detector. At a time point 0, you can see a pretty homogeneous pH distribution. The white lines in here indicate the sediment water interface line, and this is a scale bar which is around 5 millimeters. What you can see in here is that there's no steep gradient in pH right now in the sediment, and in this example or in this application, this sediment sample was then overlaid with some weathered terrestrial sediment, approximately 1 to 2 millimeters, and this caused a pH change in the region where the weathered terrestrial sediment was deployed. So you can see that the image gets more bluish, which indicates lower pH values of around pH 6.1, 6.2, something around that, and you get a pretty steep concentration gradient. And now the reworking of that deposited sediment sample was investigated over time. So you can choose from your dataset different regions of interest, for example in here from the sediment deposited area, and here the more bulky original sediment phase, and compare what's happening over time. In the deposited sediment area, you see the initial pH drop, and which is then slowly adapting back to more neutral values due to some proton diffusion processes and also reworking of the sediments. Whereas in the bulky phase, you see an initially higher pH value of around 7.4, which then slightly drops due to some proton diffusion from the sediment into the deposited sediment, but also due to some reworking processes taking place. So you can see you can record your data set, and then you can choose your region of interest, or you can also choose multiple regions of interest and compare what's happening in your sample. Sediment core samples are pretty static systems, so there is usually no flow inside the core. But the sensor foils also work in non-static systems, such as, for example, in flume tanks with constant flow or with flow at all. In here in this flume tank, from the right side, there's a flow direction towards the left side. The sediment water interface line was covered with oxygen sensor foils and then investigated for visible or for detected gradients. What you can already see in here in this respective oxygen image is that due to the flow and due to some physical dynamics happening, like upwelling anoxic pore water and downwelling highly oxygenated seawater, you get a totally different structure compared to the static systems. So you have steeper gradients, but also some surface near anoxic regions that are happening, or some deeper parts of the sediment with a higher oxygenation. In this experiment, the biodeterioration of tar balls was investigated by the microbial community in the sediment sample. If you're more interested in the respiration measurements and on the biodeterioration measurements, I can refer to the respective application note that you can find on our website. Besides physical parameters, also biological factors can alter the gradients in your sediment water samples. As for example, in this case, you see already the structures in here in the sediment, and these are burrows caused by polychaetes. The polychaetes try to transport, or they transport highly oxygenated interface near water into deeper parts of the sediment and cause an oxygenation in the upper layers of the sediment. If you now apply oxygen image to that scene, you can see in here, for example, these burrows where the highly oxygenated water from the seawater phase is pumped into deeper parts of the sediment. What you can also see is, like here, the original pretty steep concentration gradient from seawater towards the anoxic sediment parts, and you can also see that the profiles depend on where you take your measurement, because they can be highly variable depending on if you have some just plain sediment water interface line, or if you have some disturbances, such as, for example, caused by polychaete burrows. In this experiment, the burrows were then covered with some terrestrial sediment, which caused a closing of the burrows and also a breakdown of the oxygen transport into deeper parts of the sediments, causing a more uniform concentration gradient. And then after some minutes, approximately 20 minutes after depositing of the sediment, the polychaete started reworking the sediment and reopening of the burrows and causing more oxygenated water into deeper parts of the sediment again. My next example is a little bit bigger scale. This was a sand tank filled with inoculated sand sample and a capillary fringe experiment. So there's a groundwater phase, a capillary fringe, and some gaseous phase here on the top with no water saturation. And this area was then covered with sensor stripes and rasterized with the VisiSens AX systems, and then stitched together and interpolated in between to get the whole image of 30 times 80 square centimeters of what's happening in there. Here on the left side, there was an inlet where an organic model compound went in, and the microbial reworking of the model compound and respectively the respiration was investigated in this system. Here's a short video on how that looks like. So you have the inlet of the organic model compound in here. The microbia start reworking the compound, causing a drop of oxygenation because they respire. And so you have also these like hotspots of microbial processes taking place in here. For pH imaging, the values are pretty constant, and you see not much unusual things happening because the microbial respiration is not directly affecting the pH at first, but you can see a pretty distinct change in CO2 production right above the microbial hotspot in here where the organic model compound is reworked. And so you see an evolving higher CO2 concentration due to the respiration of the microbial. So usually for recording bigger areas, we designed a new type of imaging systems, the new VisiSens 2D imaging system. So you can investigate bigger sizes, but you can also investigate oxygen, pH, or carbon dioxide foils or combinations of them in one field of view with one system. So this system is compatible with all three types of sensors. It's a modular setup, so you have a variable view from medium scale, from approximately 4 × 6 centimeters upwards, and also to bigger scales like 30 × 25 centimeters maximum by adding the big area imaging kit to the system. So we can really read out large sensor foils in one field of view. Another big benefit is that you can combine, for example, multiple sensor parts or multiple sensors in one field of view. For example, different small oxygen sensors where you expect some interesting things going on. Or you can also combine several sensor types in one field of view, as indicated down here below on this image, where a sensor stripe for oxygen, pH, and CO2 was placed in one field of view and consecutively or simultaneously recorded in one experiment. So now I will hand over to Martin again for a short summary of our presentation.
Thanks again, Robert, for these really nice examples. And we are almost done. Quick summary about the two techniques we just showed. So on the one hand, the microsensors, we can sum up the microsensors measure on one dimension, so you can profile inside your sample. It can be any type of sample, and as we saw on the river, on the biofilm formation, you can also use it to monitor very heterogeneous topographies. You're only micro-invasive or sometimes even non-invasive on a very, very small scale. The profiles themselves, if we talk about time resolution, take about 1 to 5 minutes. And especially the oxygen microsensors, they show a really fast response time, meaning that if you want to measure on a certain spot, you can capture very, very fast dynamics on the seconds or even milliseconds level. For example, like photosynthetic changes, like light dark shifts, and so on, can be monitored with oxygen TS sensors. For the 2D imaging, we talk about a 2D field of view, which you can use to image actually through any transparent wall, like an aquarium or a sediment core we just saw. It allows you the direct comparison of regions and to measure inside those cross sections of a picture. The whole system can be, after being set up, be non-invasive and left there for incubations, for example, in rhizotons or growth of roots, sediment cores or whatever, for extended periods of time. And it allows you to record those time-lapse videos. So you can take an image of about every 2 seconds, and these images you can then stitch together to really continuously monitor and create the nice video images Robert just showed us. With that, we are at the end of the presentation. thanks again everyone for watching. Thanks for your time and thanks for your patience. I think we have time for a couple of questions, so I'll quickly check what questions we have here.
Okay, the first question is, can I also profile with the microsensors in soil samples, not only in sediment samples? Martin, can you give us an impression on that?
Yes, yes, you can. Soil is also possible. As Robert said before, if you do pH or CO2 measurements, the soil needs to be moist. As long as you've got 100% humidity and can actually detect pH, you can also measure it with the microsensors. Depending on the roughness or the toughness of the substrate, it depends. So the microsensors are of course limited to a certain grain size. If it gets too coarse, for example, here you could still measure down with a microsensor, but if it gets too coarse, like for example, like really hard rock we have down here, or some hard shells inside, I would recommend people to use a dipping probe instead.
Okay, and the next question is on how deep into the sediment core can I measure with the imaging sensor foils? Actually, our sensors are diffusion limited. That means that you have to you measure the interface of the sensor foil. So what you actually do is you measure in direct contact with the sensor foil, or the part which is in contact with your sample, but not into more deeper parts. If you want to go into more deeper parts or the bulk phase of the sediments, I would recommend to use the microsensors.
We have another question just came in. Someone actually would like or has samples that are actually contain some oil phase, or yeah, that have some lipids inside. So regarding that, actually for oxygen, we can say yes. So oxygen is possible. You just need to recalculate. So the measurement itself works, but you have to keep in mind that actually the calculation of oxygen is in oily substrate or oil media is actually different than in aqueous media. So that you have to take in account when you do volumetric concentrations.
But the partial pressures can be measured just as they are.
Exactly, exactly.
Okay, there's another question on possible light interferences with photosynthetic organisms. And this is related to the imaging sensors. Actually, the imaging sensors, they contain an optical isolation, which is on the sample side of the sensor foil, and this blocks the excitation light from getting towards your sample. What you can in principle do is that you can, for example, for the oxygen sensors, just peel off the optical isolation, then the sensors get more transparent and you will see more underlying structures, or you keep it on and then your sample is protected.
And our last question refers to the response time of the microsensor. I hope we answered some of that in the webinar already. Maybe to point out, so the microsensors, they are really fast, especially the oxygen microsensors. In the gas phase, they are even faster, so there we can reach speeds on the millisecond level, while in the water phase or aqueous media, we are usually on the second level up to 10 seconds. Okay, I see. This is it from the questions we have so far.
Mm-hmm. So thank you for listening.
Once again, yes, thanks to everyone for listening. We will show the last slide here. We will also show our contact details. So in case you still have questions later on, or you want to know about anything or have any inquiries about the products, please feel free to contact us directly, Robert and me, Martin, yeah, directly via the email address. And thanks for watching again. and have a good day.
Have a good day. Goodbye. Bye.